Great, yeah. They'll not understand the beginning at all. No. One of the major challenges we have despite all this progress is that still an appreciable amount of patients who come in with rare diseases do not get a genetic diagnosis. So, upwards of about 50% of individuals, we might be able to provide some insight into what's driving their disease. But there's still this large subset where there's no candidate variant that's detected by whole exome sequencing, and another subset where there are suggestive variants of unknown significance, but we still really don't know what's driving the disease there. And so really thinking about how we're going to push the field forward, we're starting to think in this consortium of what are the types of things that will catalyze the next paradigm shift so that we can get more patients coming in with conclusive genetic diagnoses. So GREGoR is funded by the NHGRI. It has a major goal to significantly increase the number of Mendelian conditions with identified genetic causes, but then also to work with patients and individuals who have previously had exome negative results or negative panel results to think about what types of technologies would provide a genetic diagnosis. And so really we want to focus on the subset of Mendelian conditions for which a candidate gene may not have been identified using whole exome sequencing alone, or even when that gene is known, the underlying genetic diagnosis is missing. And this is sort of branched into 5 sort of high-level major goals for the consortium. We obviously want to shift the paradigm again in similar ways to how exome sequencing was effective, but it's very hard to predict paradigm shifts. So we're trying to think about what are the things that facilitate progress. We want to increase the diagnostic yield. We want to do that not just for patients here, but for patients everywhere. We want to establish and provide the best practices for working with a range of different omics data in clinical settings, and also create a data set of broad utility. So I think one of the major challenges that has existed so far is that a lot of patient data and genomics and multi-omics can be siloed in different locations. And if we can provide a resource that allows people to access this data more effectively, it will stimulate the development of new technologies and computational approaches. And then finally, we want to make rare disease diagnosis equitable. So if there are things that we can do that can reduce the cost of technology, improve where it's deployed, we think that will just have a greater global good for this type of work. So who is GREGoR? We are one site, so it's the co-PIs of the site are myself, Matt Wheeler, who'll speak next, and John Bernstein. We're also benefited by groups at Broad, UCI and Vitae, Baylor, University of Washington, and there's also a data coordinating center at the University of Washington, which is working to bring all this data together and provide it to the larger research community. We're already benefited by the fact that there has been a number of exceptional papers and lots of work out there describing novel approaches to solving exome negative or panel negative cases. And this is just an example from a review paper published here at Stanford by Shruti and Ewan, showing that there are a range of different non-diagnostic exome, there are a range of different approaches for non-diagnostic exome sequenced individuals. And those can range from looking for structural variants to integrating multi-omics to look for outlier effects, to periodic reanalysis of exomes, metabolomics, which is actually a major focus, and Tony alluded to a great conference coming up in that area, to other complementary types of omics. Once we get all this data, you can actually funnel it down and start thinking about integrated analyses. What do you learn from these different components of information for different patients? But beyond that, even when we have variants, to get it to the standard to actually say this is something pathogenic that's important for a patient, we also need to think about the technological approaches that support the functional follow-up of these types of effects, whether it's a gene or a variant. So really our thinking in this consortium is that to shift the paradigm, it's going to involve being able to effectively use this combination of genomic technologies that are present in the field, and thinking about for which patients, for which variants, for which genes can we successfully deploy, whether or not it's a transcriptome or metabolome or CRISPR data or anything else, zebrafish, knockouts to actually get tractable information that helps that individual. So the aims of our specific site hopefully support, what are the goals, these major goals. So we want to recruit to the Stanford GREGoR site somewhere on the order of 400 prospective families and 100 retrospective families, mostly people that we've worked with through the UDN and their immediate family members. This is our baseline aim. We obviously hope to go much larger as we get better with the technology, as there's reductions in cost over time. And we're focused on people where there is prior exome or genome sequencing. They've been comprehensively phenotyped, but there's no genetic diagnosis so far. There are a range of different goals that we have. We want to generate a lot of multiomics on each patient and their immediate family members. We want to deploy innovative computational approaches that are being developed here at Stanford and elsewhere. And we also want to think about what are the advanced functional validation assays that we can bring to bear to understand variant effects and specific genes for these individuals. So this is the organization of our center. We're benefited by a large number of really outstanding co-investigators, complementing, from genomics to computational biology to functional validation, molecular biology. This is not even fully representing the amazing clinical genetics team, medical genetics team that has been working as part of this project as well. We meet regularly thinking about how we can intersect all of this great research here on campus with the clinical practice for rare disease research. And we've already started recruiting people. So as I mentioned, this started just a year ago, so there's been a lot of work to sort of get it up to speed, to think about what are our recruitment strategies, what are the types of consents, how are we going to bring people into this consortium. So you may have seen this type of poster already being sent out, looking for participants affected by undiagnosed rare diseases. So if you know people or you're part of a research study and this seems interesting to you, I suggest maybe you might want to contact us and see if there's opportunities to work with us there. We're specifically interested in cases with suspected Mendelian disease, maybe where the gene candidate is already identified, but both parents available for testing as well. So this is our planned enrollment over the period of time. So we're somewhere in this 2023 phase, hoping to have 160 people by the end of 2026, over 400 different cases. Our current enrollment looks like this, however. So we have 100 cases in the pipeline. 64 prospective cases have been referred. 36 retrospective cases have voiced interest. We've got about 37 people enrolled so far. So there's still lots of opportunity to expand. And we consider this early days, but it's been exceptional to see this grow week to week, people coming forward and seeing the opportunities that our site may provide them. Our recruitment sources are mostly focused around the Bay Area, Stanford as a primary hub. I was just giving an example of some of the places we've reached out to for community referrals as well. Over time, we want to expand this even further. So we've been working with translation services to have a Spanish translation of our online referral and application. I mentioned we launched the patient recruitment in May 2022. We've launched sample collection and been working with CTRU since September 2022. And we're continuing outreach in a lot of different local health centers with actually a hope to be able to work with UDNs across Latin America as well. This is just some insight into our current patient demographics. So it's mostly split female male, it's split pediatrics and adult, and there's a range of different diseases. So we're not specifically constrained into cardiovascular disease or neurology. We're thinking about how this technology can come to bear for all sorts of different patients with different indications. So what kinds of assays are we performing? So when a patient comes in, we want to have complete multi-omics profiles on them, plus their parents. We're doing whole genome sequencing. This is both short read genome sequencing, long read genome sequencing. For a subset, we're planning to do optical mapping as well. There'll be RNA sequencing on accessible biospecimens, through methylation sequencing, attack sequencing, metabolomics. And again, you can see the team that's been involved with trying to set that up. So already some of these individuals, we have this type of data that's accessible. So you can see there's an appreciable proportion where we have exome sequencing. Most of them have had that before, or whole genome sequencing. There's about 20% where a candidate gene is previously known. So in many cases, we still don't know what the candidate gene is. Most of them have had transcriptomics, subset with metabolomics, and I'd say about a third have had long read whole genome sequencing. The goal, though, is to complete these matrices on all of these individuals. This is just a snapshot of our tissue analysis plan. So when someone comes in and they get sent to CTRU for collecting biospecimens, we get PAX-gene tubes for blood, EDTA tubes for plasma, CPT tubes for PBMCs. We'll potentially do skin biopsies if there's good evidence that that will be useful. And we're also slated to generate IPS. So we've got a subset of patients that we can that we're able to generate cell lines for. So once we get all this information together and these biospecimens, part of this, major aspect of this consortium is to accelerate data sharing from all of the research centers to the larger research community. So part of the last year, we've been working within the consortium to really think about what are the data sharing pipelines that we can put in place that will make sure that this information is maximally used. So this is just, again, a snapshot of what's currently happening. The goal is that every quarter, data will be in a bucket that will be uploaded to the DCC and then made available to the broader research community. And this involves everything from sample intake to assay, internal QC, having analysis ready data, uploading files to the workspace, validating data, and then setting these workspaces to read only so that the DCC can import these data. So we really want this to be a seamless, accelerated process for people coming in here. And there are multiple challenges that everyone faces when they try to do new technologies at scale rapidly for individuals. Our external data sharing policy is designed to be concordant with the current GDS, which actually was just updated about a few days ago at the NIH, to allow this type of external data sharing and to be concordant with their expectations. Again, it's going to have rapid data sharing. We're going to be plugged into a lot of different external data repositories. So not only just our public-facing website, all this data will go in Anvil, which is a cloud-based solution that people can access and work with that data. They don't have to pull it down to their own system. As long as they have cloud credits, they can do analysis there. But then we'll also make sure that there's the appropriate linkages to Matchmaker Exchange, ClinVar, and when relevant, Model Matcher, Variant Matcher, and IGVF. And some of these are already in place from the previous CMG program. So I mentioned this is deposited quarterly to Anvil. So Anvil is the National Human Genome Research Institute's genomic data science, analysis, visualization, and informatics lab space. It's actually a really amazing idea that instead of everyone trying to download copies of the data can exist in some location with a well-specified data model, and you can actually log into the systems and work with the data there. So because we have this as sort of a foundation, and we've been thinking across sites about what is a data model that concords across all the different activities that are being done, there's the possibility to actually generate much larger, uniform, harmonized data sets from patients across the entire consortium and share those results quite quickly with everyone else. Just some other features of our site, which I think are exciting and things that people might want to plug into, is that we really believe that accelerating data sharing here is going to help us build new computational approaches and bring exceptional researchers to exciting data in the rare disease space, so that they'll be able to have the capabilities to do that. So I think if the data is all firewalled and behind protected infrastructure, it's hard for people to want to do analysis on it or think about building new methods here. So our hope is that this helps accelerate that cycle of building exciting new methods here. And these are just a subset of the investigators that are starting to think about how to work with that data in different ways, from using deep learning approaches and multi-omics for variant effect prediction to integrating genomic backgrounds on top of for variant effects and understanding about the combinations of those and how they might lead to a particular outcome. We also, as I've mentioned, we have lots of opportunity for collaborations around functional validation. So some of the co-investigators in the group, Michael BASIC is one example, has been doing CRISPR screens. We have MPRAs that are set up. Tom Kutermis has CRISPR-I mouse models. And again, we have the capabilities to generate iPS cells on a number of these patients in a range of different developmental contexts. Beyond just what we're doing here, GREGoR actually has a much larger ecosystem for doing variant to function types of assays and deploying different technologies. So there's a working group that's part of it called FONT, which actually involves membership from all the different sites. And they come together to think about, what are the genes we want to go after, what are the variants that we want to go after, and what are the best technologies that are available to do that. And so people can actually propose, different assays and different genes. I'll just give an example of that. So one of the first steps we did as a consortium is actually think about what are all the technologies that we can bring to bear across all these different sites. And this is actually just showing the list of functional assays. Some of them are discovery-based, some of them are functional follow-up based. But you can see with over 40 different assays, there's a lot of different opportunity for doing different work with this consortia. However, there are also different challenges and limitations that we're hoping to be able to address given these range of assays as well. And those go from the fact that we just get so many candidate rare variants, particularly when we consider the non-coding genome. We're also starting to think about how do we go after complex genetic etiologies. So if you have something that's digenic or oligogenic, like what is the functional assay that we can use there? What types of information gets us towards selecting and sharing target genes and variants more broadly as a research group instead of within specific sites? Selecting specific model systems, whether or not they're in vitro or in vivo, and then eventually moving technologies to clinical application. I think all of us will probably recognize this here. There's so much incredible advance in genomic technologies, but the number of those that make it into a clinical setting is actually quite scarce overall. And so what is preventing just getting RNA-seq into the clinic or metabolomics into the clinic more generally? Let alone many of these other different types of assays. And so we're starting to think about how do we get these things there? How do we prioritize them? So I think it's a very exciting aspect of this consortium. And so just a couple of last points I just want to show you, given where we're at. We do have this gene technology assay nomination form. So people can come in and they can say, this is a gene I'm interested in. I've submitted it and it gets discussed and nominated to this ClickUp form where we'll think about the types of functional assays that we'd want to deploy on that gene or for those different variants. So I think this is trying to harmonize activities across the consortium and to go at where technology is going to pay off in different contexts. And then there's a range of other opportunities. So for those that see this as something that they would like to be involved with or connected with, it's everything from referring patients you might be working with to samples you may have access to using some of these computational or experimental techniques, to even thinking about how do I rapidly share data with the community. I think these are all components of what the GREGoR site is hoping to address and provide to everyone here. So with that, I just want to thank the team. It's an exceptional group of people to work with and encourage you to contact us if you have further questions. And if you're interested in more of the nuts and bolts and the science that we're doing inside the consortium, we do have these biannual site-wide meetings. The next one is coming up January 25th. And so there you get to see a lot of the different projects and hear about different cases we're working on. And they're always really exciting. So thank you. Yeah, no worries. Yeah. Next up is my esteemed colleague.
Excellent. Well, thanks, Stephen. And we're really excited about GREGoR Stanford site, and I think certainly we've learned a lot over the past 10 years, and some of that learning has been from the Center for Undiagnosed Diseases here at Stanford. And so I'll talk about this Center for Undiagnosed Diseases, a little bit about the Undiagnosed Diseases Network as a whole, and also about a case that I think illustrates some of the challenges and opportunities for helping patients with rare and undiagnosed disease. So the first time I gave this portion of the talk was February 28th of 2020. I don't know where, you may not remember where you were that day, but 2 weeks later, many of us remember where we were when kind of the whole country shut down. And so imagine for a moment, this is your thermometer and you have a fever. And perhaps you've just been at a conference where a guy came from Italy to chat with you. And then you read the news the next morning. And so, if this was you, would you be undiagnosed? Yes, but chances are it's one of these things or maybe this thing. But nowadays we kind of know what's going to happen. Maybe you not feel well for a few days. You need to stay home. You'll take one of those COVID tests or something like that. If you're unlucky, it's this. This is a biopsy of a lymph node from somebody who had Hodgkin's lymphoma. But for most people who've had this happen to them, then they go to Dr. Google. Well, this may not work. Oh, yeah. Unfortunately, Dr. Google got lost in email. But what you would see is within 10 seconds, you can get advertised to by the Mayo Clinic or Stanford or other people who can tell you how to treat Hodgkin's lymphoma, what the new technologies are, who the best doctors are, et cetera, et cetera. This patient is not so lucky. So he starts out as a 6-month-old with an empyema, a pneumonia, and a fusion that's infected. Not super typical, but one possible outcome after your 6-month-old has a fever. At 18 months, has had recurrent episodes of ear infections. Again, like not that unusual. Many kids get ear infections. Some of them need ear tubes and things like that. Become best friends with their local pediatric ENT. At 4 years old, he had joint swelling in multiple joints and a non-traumatic fracture related to this. This is now beginning to get into the area of this is not something you would expect from your average 4-year-old. And so at this point, this patient started being seen by several specialists and really didn't have an answer for why this was happening. At 5, he'd had multiple episodes of bone infection. And 4 years later, multiple joints with a destructive bone infection type picture. And so this is an example of somebody who's really undiagnosed. They have many symptoms, many clinical findings, many notes in their chart, but no one at this time was able to say, this is the thing that's driving all of these findings. And so there are many people across the country who are undiagnosed, and you can be undiagnosed in any sort of disease spectrum. Most undiagnosed patients don't know their prognosis, settle for empiric therapies, live with chronic uncertainty, have fear of the unknown. They worry about themselves and their family members, often wonder what they did wrong or what their parents did wrong, and often they have a rare disease. And so the Undiagnosed Diseases Network and the Undiagnosed Disease Program beforehand has really come together to try to provide a central way for patients with undiagnosed diseases to be evaluated and to try to move forward so we can answer some of these questions. Work from the Undiagnosed Disease Program and from genetics and lots of other parts of medicine have shown us that both genotype and phenotype can facilitate diagnoses. And so in distinction from the GREGoR Consortium or the CMGs, really, the UDN's goal is to generate both genotype and phenotype information to help facilitate diagnosis. And that's been kind of the goal from the start. The phenotype that you see and the phenotype that you collect may have different levels of importance at the clinical side. But when it comes to diagnosis, even some relatively benign or incidental phenotypic clues can be key to determining a molecular diagnosis. And so this patient had a few other key findings. They had a pneumothorax and pulmonary hypertension, early post-delivery, delayed tooth eruption, elevated immunoglobulin E and eosinophilia that was consistent over time, and did have some empiric therapies that may or may not have contributed to some of the other symptoms he had. He'd had laboratories, and this is a brief subset of a extensive panel of testing he'd have over the years, but had molecular testing for two syndromes that it was felt that many of his findings were similar to, and the molecular testing was negative. And then at the time, he underwent exome sequencing that's clinically available, and that was basically completely negative. So you get a report that says, we sequenced this patient's entire exome, and we have nothing to report to you. That's not the same as we sequence your entire exome and there's nothing there. And so that's where both UDN and GREGoR and other in the CMGs and other Mendelian research consortia, really start looking. And in 2008, when the Undiagnosed Disease program started at the location on the right at the National Institutes of Health, exome sequencing was just emerging as a technology. And so part of the impetus for bringing together the Undiagnosed Disease program was actually related to multiple individuals getting exome sequencing data and not knowing how to analyze it in an efficient manner. And so that helped nucleate what became the Undiagnosed Disease program, which continues today and is continued to be funded as an intramural program. In 2013, when the Undiagnosed Disease Network was formed, it was clear that the NIH clinical site alone couldn't do the work that was needed to help undiagnosed patients across the US. And so places like Stanford applied and became part of the Undiagnosed Disease Network. And now there's 12 clinical sites that have been operational for the past 4.5 years, another 3 or 4 that are coming on board this year, as well as the legacy site at the NIH clinical site. There's also undiagnosed disease centers and networks outside of the UDN across the country and across the world that have really used a similar model of collecting phenotypic information and genotypic information to help identify causes of rare and ultra rare diseases. And so this is a paper from 2012 or 2013 that kind of describes how the UDP works. Again, it involves in-person clinical phenotyping, multiple specialists, typically agnostic to age of the proband or individual of interest, and a single research protocol. So if you have a patient who has an immune abnormality and a patient who has a cardiac abnormality, they sign the same consent form, their samples go in the same place, the same people look at the data. And that was really one of the key innovations that made it practical and efficient. And there at the NIH, which many of you know, there's a S in National Institutes of Health. They don't always work together very well. And so one of the key things that the UDP was very successful in is facilitating data sharing across institutes. Stephen mentioned Anvil as NHGRI's version of this. We work with a different group that uses a common funds data sharing ecosystem, and there's ones for other institutes, and still it needs some optimization. And so this is a map of the UDN as of the beginning of last year. There's now 3 additional sites that will be added to this map in the next few months. But ourselves in UCLA have been representing California since 2013, 2014. And we are continuing in operations. To date, the UDN has received almost 6,200 applications. We've evaluated about 2,100 participants, and another 300 or so are currently in the evaluation process. And overall, we've diagnosed with a sort of firm or very likely diagnosis about 630 patients. And there's a substantial number, and our estimate from our own site is about another 600 patients who have a suspected diagnosis, where it's working with scientists and collaborators to try to confirm those diagnoses. And so there's significant work still to come. Our center has a number of team members. At the center is a patient and their families. There's research coordinators, some of whom are here today, and genetic counselors who are really our key workforce on the ground that interact with many of these other individuals, principal investigators and core clinician scientists who help direct both inquiry and clinical evaluation, and also a team of informaticians and technicians who help with some of the wet and dry lab work we do. And we have collaborators in both one-off and consistent collaborators across many of the specialties across the campus. And so when we think about, again, a patient at the center, what can we do? Well, we have a clinical evaluation. This is evolving over in part because of the pandemic. It's accelerated our uptake of telehealth to facilitate evaluating people across the country. And we do some standardized phenotyping. So we collect the same information on everybody, but we also are phenotyping specific for particular disease phenotypes in detail on a research basis, and sharing of that data across the network through a central data portal. Almost everybody, but not everybody, ends up getting additional sequencing. So we're doing genome sequencing on almost all of our current patients, performed through a clinical lab. So we get a clinical report and have a clinical sample for validation. We also do a number of research sequencing. And so some of the work that's led to our success at setting up the GREGoR site and transcriptome sequencing with Stephen's lab, long-read genome sequencing with you and Ashley's lab, and Stuart Scott in the clinical genome program, and short-read genome sequencing, mostly with collaborators today, but also with the clinical genome program going forward. Importantly, as I mentioned earlier, there's the exome, and there's a report, but there's also the data from the exome. And so we do spend some effort on reanalyzing that data with an eye towards finding a rare disease that potentially has never been described before. The UDN also has a model organisms screening core. This has been in existence since 2014 and expanded in 2018. Now in fly, fish, and worm, and then a series of collaborations actually with what used to be the comp mouse engineering facilities. And so there are a couple of different avenues to go in that direction. And then there's also external collaborators in the larger model organism community, where the UDN and other networks like it have some sharing and sort of efficiency optimization of making model organisms and sharing those. And so this has been really instructive for many cases that have been solved, particularly for new disease genes. And then we do some other research both here and across the network, and we particularly leverage our colleagues with the Human Immune Monitoring Core to do immune profiling in many of our patients who seem to have an immune phenotype. The metabolomics cores, both through the UDN and through Mike Snyder's group. And then we do cell-based studies in the wet lab. And one of the new things that the UDN has sort of really been an evolution about over the past 2 years is moving towards precision therapeutic development for, again, those 600 new patients with new diseases or new diagnoses, and trying to figure out ways to make that scalable, which is not a trivial problem. For the average applicant, this is kind of the expected timeline. Everybody gets an application review and a decision about whether they're appropriate for our research protocol. There's questionnaires that we give them. And then for people who we're bringing on site, clinical evaluations, and then we do omics profiling kind of in a stepwise fashion now. And then we bank samples and tissue as appropriate for future use and for sharing. And there is a UDN-wide biorepository. And so samples from each individual across the network get stored there. And if in the future we want to look back at somebody who was seen in 2014 at Vanderbilt, then we can ask the biorepository for those samples, and that can be helpful as well for new disease discovery. So this is an example of a patient's chart who is seen at the Center for Undiagnosed Diseases. Fortunately, most patients end up sending us electronic records now, and so our coordinators have a bit less time opening and closing mail. But there are people who have literally years of medical data that our team sifts through. And this is really the beginning of our evaluation, looking through that chart, pulling out the pertinent phenotypes and any genotypes that may have been found. In this case, the patient who presented initially at 6 months with a fever, we did reanalyze the exome sequencing using an automated method, and then went to whole genome sequencing and still had no clinically significant findings. We did a clinical genetics evaluation, and there was a concern on a review of a skeletal survey that there may be craniosynostosis. Before we get there, there's these are some examples of joint X-rays at the time of analysis, and so I hope that neither you nor any of your family members ever has a knee that looks like that. But basically total destruction of the joint as a consequence of inflammation and infection. And so when we looked at the head CT, what was noticed is that there's a loss of suture lines, craniosynostosis, and this wasn't appreciated before this UDN meeting. So again, sort of an astute clinician, appropriate testing, and then finding this and a few other phenotypic findings. We did cytokine profiling through the Human Immune Monitoring Core and found greatly elevated IL-6 level in comparison to other cases, including other patients who also had immune disorders of uncertain etiology. And then we reanalyzed exome sequencing with a few new pieces of information about phenotype, as I just mentioned, and came up with this variant, which is in the IL-6 single transducer, also known as GP130. And this was a variant that had been previously seen, and there's a known disease association of immunodeficiency and craniosynostosis, but it's autosomal recessive, and this was a single variant. And so one of our former genetic counselors went and looked in, at the raw data in a program called IGV, and saw this. And so with the red arrow is the variant we expected to see. And then just a few bases to the left, so each of these on the right, each of those columns is a single base pair being called. If it's gray, it's identical to reference. The colored variants are, in this case, heterozygous in 2 locations. And what she noticed was every read that had the G here did not have a G upstream a few bases, and the converse was true. And so this is consistent with biallelic or biparental inheritance of 2 variants in trans, 6 bases away in the genome. And for some technical reasons, this red variant was called by at least 3 independent labs variant callers, and none of them called the 1 6 bases upstream. And there's actually good statistical reasons why that happens in terms of reducing false positives. But we also noticed that there's another variant downstream that helped us phase and make sure that these were real variants. And so what we find is actually this is 2 variants in the same gene. This now fits the autosomal recessive model. But this second variant was never seen before in anyone who'd been sequenced at the time. And so we didn't know, again, if this is something that's cryptically common, but it did have evidence through some prediction programs that is likely to disrupt splicing. And so we went to transcriptomics through Stephen and Laura Frassard, who's a former postdoc who's now working at the NBT site of GREGoR, and use RNA-seq to show, in fact, that second variant was causing splice disruption. And so now we have a molecular evidence of a splice disruption. We have clinical evidence from other patients that this other variant is potentially disease-causing. And now we have what looks to be autosomal recessive disease in this gene. We collaborate with somebody at University of Oxford, Dr. Ulig and colleagues, who were doing functional studies of this gene and confirmed that our patient's fibroblasts basically didn't respond to IL-6 appropriately. And so at the time, there were 2 other case reports that our patient joined a couple others, and there's been more since then. But this is sort of fit the mechanism, the expected findings, and other clinical findings of other patients. And so this is a diagnosis. So as I mentioned at the beginning, we have across the UDN seen over 2,100 patients. There's still 1,500 of those patients that are undiagnosed. Our goal over the past years has been to transition out of the common fund, which has both benefits and risks. It's congressionally mandated to fund projects for 10 years and no more, a day longer. And so in the congressional omnibus bill that was passed just before the holidays, they have a line item that will continue the UDN, at least for this budget period. And the goal is for NINDS to help us continue the project. In addition, Megan Halley has been working with many of our colleagues across the country to set up a foundation to support the UDN long-term and to help patients and their participants. And it wouldn't be without many, many people at Stanford and across the country that we'd be able to do this. Again, there's many patients still where we're using other approaches, using resources like GREGoR, using other resources through HMC and other projects to help move things forward. MCHRI has been a tremendous partner for helping spin up some of the new technologies that we brought into UDN over the past 10 years, and we'd hope to continue to partner with MCHRI for the next 10 years as well. So thanks to many of you online and in the room who've helped over the years, and we're really excited to start a new chapter for the Center for Undiagnosed Diseases. So.
We'll take questions from the audience here, and then also for those online, if you want to put them into the chat, we can also take them there.
Just refer everything to you.
So you have 1,500 undiagnosed. What fraction of those do you think GREGoR can solve?
Yeah, I think so. What we find is that there is probably 25%, maybe 15 to 25% of patients where there's probably an environmental trigger or infectious trigger that are very hard to track down definitively. We have a few cases where we've been using antigen discovery platform to try to identify essentially super rare autoantibody as a mechanism of disease. We have kind of like one case where it's pretty clearly that's the answer, but I'm not sure we're ever going to be able to prove it. And then a few where there's something that it looks suspicious, but I think to convert that into a mechanistic story is hard. It's hard even for actually ITI just announced one of the people who was working on the multiple sclerosis project is now a faculty member here. So we're hoping to learn from that experience, but that was hard to prove because of the epidemiology. And I think for rare cases, it's hard to prove as well. But for the other proportion, I think a good percentage. So we know from UDN that just going from exome to genome, you get maybe 8 to 10% additional yield. With transcriptome, 10 to 25% yield, depending on who you talk to and what you've looked at. Recapturing variants that neither exome or genome standard pipelines collect is something that we've spent a lot of time with over the years. That's maybe another 5-10%. And then I think things like the telomere to telomere build of the genome and some of the technologies that the new sequencing technologies allow you to discover or identify variants that you can't find with short read. That's, I think, more unknown as terms of what percentage that is. And I think from my day job as a cardiovascular geneticist, it's also clear that there's a substantial proportion of polygenic inheritance that goes into certain phenotypes. And so it may be that those will be on an individual basis difficult to solve, but collectively we will be able to generate sort of risk approximations or use some of the transcriptomic or other omic technologies to say this is the pathway that's disrupted. And that's been a sort of collective vision of ours that really hasn't borne out yet, but we're hopeful that will contribute. So, we'd like to say all of them. I think, we know from the people that we haven't yet diagnosed but are on the path that it's roughly 50-50. That we have, for every patient we solve, we have one strong candidate that's somewhere in the pipeline.
Just wondering, so many syndromes have multi-tissue readouts and complex phenotypes also developmentally appear differently. And a lot of lesions you have may not be treatment tissue specific. So is there a pipeline or thinking about how to decode the tissue readouts and whether a reference data set from known Mendelian diseases, things like obviously thinking about celiopathies, right, where the template helps us a lot to map complex phenotypes back to a concerted number of mechanisms. How do you deal with tissue specificity?
Yeah, I think there's a lot of different components to how we're hopefully trying to address that. Oh, so the question was how we hope to deal with tissue specificity of different types of rare diseases. So there's a lot of different pathways to how we're trying to address that. The first one that comes to mind is we're actually part of something called the Developing GTEx Consortia. So this is looking at earlier individuals across a range of different tissues, what types of cell type expression and changes manifest through basically ages from 0 to 18. I think we're starting to see there are single cell data sets that go earlier into development. And so these give us some insights into which genes are active when, or which regulatory elements are active at different points. And so we can start to think about what's the context of variation with respect to the activity of those types of elements through those types of maps. That's definitely one point. We're also thinking a little bit about what can we pull out of samples that we can easily assay from a patient that allows us to better triangulate things that might, be happening in harder to access tissues. And so obviously IPS is one amazing example of, you can get some cells, you can transform them into different cell types, but it's also a labor intensive and expensive process. And so thinking about how do we get, proxies to that, or are there places where, we can modify library preps in different ways or go deeper in different ways to understand splice variants, for instance, that might not be, that might be sporadically expressed lowly in one particular tissue, but highly expressed in another, that give us insights there. So I think a little bit of how we leverage the technology in new ways will allow us to address some aspects of tissue specificity as well.
I mean, it seems just to follow that up. If there was a universal surrogate tissue, that would be wonderful.
Yeah, actually,
you can get something out of surrogate tissues.
Yeah, and there are efforts to try and generate things like that. There are obviously organoid systems, but there's embroid body systems as well, where you can couple those with single cell assays. So it really depends on how much you can do that at scale to expose, all the dynamics and the temporal aspects of what's happening there. And so, if there are smart ways we can approach that, we will, and we're thinking about it. Yeah.
Questions?
Oh, sure. Yeah.
One from Julie.
Yeah, so just one other comment to that. What's been extremely helpful for the UDN is to have in our hands what genes are actually expressed in the samples we get. And so to say like, oh, IL-6ST, we can do that in blood, or we can't do that in blood, is super helpful. And so we can ask that, like with the prep we currently use, can we get it in blood, fibroblasts, IPS? Do we have to go to embryoid bodies, or do we have to go to the cell of interest? And in some cases, we've been like, it's only in skeletal muscle. We're going to make some IPS-derived skeletal muscle.
So Juliet's question here is, patients that are negative in UDN or lacking a diagnosis, can they go to GREGoR? I think the answer is yeah. Yeah, we hope for sure, definitely. So reach out and contact us, and we can see if they meet the inclusion criteria there.
Yeah, and we have been going through our existing UDN patients where we feel like we've kind of reached the end of the UDN odyssey without a diagnosis and recontacting people, but there may be people more recently where we still feel like we're in process in UDN and they haven't yet been kind of referred for GREGoR.
So the next question is from Darius asking, are you following up with your subjects to see how the diagnostics improve their treatments? So and maybe you want to take a UDN perspective.
Yeah, so from a UDN perspective, there has been a couple of network-wide initiatives to look at the impact of diagnosis on treatment and treatment decisions. It's definitely not consistent, but there are many patients who their treatment has been dramatically altered by having the sort of right molecular diagnosis. Actually, the case I showed, there's various times where they're being treated as immune deficiency versus autoinflammatory disorder and then infections. And so I think like knowing where in the mechanistic cycle to stop things is important. And then but there are other patients where we know what the genetic diagnosis is, and it's fairly obvious that there's no clear mechanistic therapeutic easily available. And those patients are being evaluated in the UDN space through the therapeutic matching committee. And then we're working with industry and academic partners to determine if they might be appropriate for sort of a gene therapeutic approach or ASO approach or a drug repurposing type approach. But that's again, something that the UDN is emerging into. I think there's things that Daria's program and Spark and Stanford could likely teach the rest of the network, and also places where therapeutic development is challenging at any scale, but particularly when you're thinking about very small numbers of patients, you know, the cost has to be manageable. And so I think that's something that, again, is something that the UDN investigators, along with a lot of other people who work in the rare disease space, are trying to grapple with, is how to do this in a not completely inefficient method.
Great. Well, because of time, we should close now. Thank Matt and Stephen for the great presentation. And thanks to the audience for coming. Just wanted to then remind you, the next demonstration symposium is Monday, February 6th with Natalia Gomez-Espino at noon. So please sign up for that. And we'll see you then.
Oh, yes. Yeah, so we can yours, so we want to, like, at the end of the day, harmonize, establish